This is Leonard Mazur. I'm the Executive Chairman of Citius Pharmaceuticals, and I want to thank everybody for taking the time to meet with us. Preferably, we'd like to meet in person, but this Zoom substitute is the way of the world today. So, first of all, just to make sure that everybody is aware of the full disclosures that are associated with a publicly traded company, we strongly advise you to consult with sec.gov, where we have all of our filings. And for full transparency, it's very important that all investors or anybody contemplating an investment be fully informed of all the events at the company, which we have fully disclosed on the sec.gov website. So the company, to give you an overview of the company, is that we have 4 active drug programs with one that's in a late stage. It's our lead product. It's Mino-Lok, which is an antibiotic lock solution. It's had a favorable review from the Drug Data Monitoring Committee this past in the year. We think that we have something here as a result of their readout that said that we should continue on with the trial, and they requested another readout, which I'll cover for you a little bit later. The markets that we address are large. We feel that we're addressing markets that have unmet medical needs. The catheter-related bloodstream infection market, which is served, potentially will be served by Mino-Lok when it's approved. It's estimated at $1.8 billion worldwide. Presently, there are no drugs in that market segment. ARDS, which is acute respiratory distress syndrome, is another market opportunity for us with a drug that we just recently licensed from a biotechnology company in Cambridge, Massachusetts, for ARDS associated with COVID-19. We also have a drug that we licensed from MD Anderson to prevent tissue expander infections in breast cancer following breast cancer surgery. This market is somewhere around $500 million. And then last but not least, we also have an entry in the prescription hemorrhoid market, which is totally unserved today with prescription drugs. There is no prescription drug approved by the FDA for the treatment of hemorrhoids. And I'll cover that in a bit. Our management team is unique in a lot of ways, in the sense that we have a lot of expertise in both M&A as well as on the science side, and as well as in certain therapeutic categories where there's a lot of experience within the company. What separates us out from the rest of the pack, as we like to highlight, is that the founders have invested $26.5 million into the company. That's not with stock options. That's with direct cash. We have gone in side by side on a significant percentage of the raises the company has done. This is critical for your knowledge, because it really is testimony to our belief in the company, our belief in the technology, and a belief that we will succeed and we will have approvals. I get a lot of questions all the time about where does that name Citius come from? Citius is actually part of the Olympic motto, Citius, Altius, Fortius. Citius is faster, and hopefully once we get approved, we'll be Altius and Fortius, higher and stronger. So in terms of the management team, this is for all of you to see, and you can certainly consult our 10Ks and look at our backgrounds much more in depth. But suffice it to say, myself, I've got a combination of big company and little company and startup company experience over the years. Had some very successful exits with companies that I co-founded or founded in the past. Myron Holubiak, who's our CEO of the company, is the former president of Roche Laboratories here in the U.S. and is considered to be one of the top antibiotic marketing minds in the pharma business. Jamie Bartushak is our Executive Vice President and Chief Financial Officer. Had over 20 years experience in corporate finance, M&A, and strategic planning, and has worked extensively with myself and Myron over the years. Our recent addition to our staff is Myron Chwichman as our Chief Medical Officer and EVP for the company. Dr. Chwichman joins us from Celgene, where he spent 5 years as the Vice President in charge of their lymphoma program. Prior to that, he spent 23 years at Roswell Park Cancer Center in Buffalo, which is one of the leading cancer centers in the United States. And while there, he co-published more than 180 peer-reviewed journal articles. So he truly is a KOL in his own right. Gary Tallarico is our EVP Operations, extensive background in terms of corporate expansions and startups. Alan Lader is our VP of Clinical Operations, formerly an instructor of medicine at Harvard Medical School. And Andrew Scott is our VP of Business Development and our face to the street. So the Scientific Advisory and Medical Advisory Board that we put together is second to none. Part of it is led by Dr. Issam Rad, who is the chair of infectious disease at MD Anderson, a hospital in Houston, which is considered to be probably one of the top cancer treatment hospitals in the world. He's authored many papers and is the inventor of our lead drug, Mino-Lok. Dr. Rupp is well known as well in the infectious disease community. He's actually the one who treated the very first Ebola patient in the United States. And Dr. Zmermal is also well known in infectious disease and has co-authored many publications, as well as co-authored the guidelines for dealing with the prevention and management of catheter infection. Doctors Selber and Viola are our consultants and scientific advisors on our Mino-Rap drug. They're also at MD Anderson in Houston. As I pointed out, that we have recently licensed a cell therapy drug from a company in Cambridge, Massachusetts. The scientific advisory board that we have there is led by Dr. Michael Mathey from the University of California in San Francisco. Dr. Mathey is considered to be a world-renowned expert in mesenchymal stromal cells, as well as in other areas related to acute respiratory distress syndrome. The other members on that board, Drs. Levy and Ware and Engbacher, are also well known in their respective fields. So let's take a look at our pipeline. Our pipeline is, as you notice, has a lead product called Mino-Lok for treatment of catheter-related bloodstream infections. It's in phase 3, as you can see. If you're not blocked out by the little screen that has all our pictures on it, we're just about through phase 3 at the moment. And we also have Halo-Lido or Citius-02. That's our hemorrhoid drug, which is about to enter in phase 2A. And then we have Mino-Rap and the mesenchymal stem cell drug, which we term Citi-401. And now these are at the IND or pre-IND level at the moment. So let's go into Mino-Lok, our lead asset. Mino-Lok is a very unique product. It contains minocycline, which is an antibiotic, sodium EDTA, which is a chelating agent, ethanol, which is alcohol. It's a patented combination and has a composition of matter patent as well as a formulation patent. This is for saving or salvaging catheters that cause bloodstream infection. So what happens is, and this is a schematic of the various types of central venous catheters that are out there. Most of the time, these catheters are inserted surgically, and it's mostly cancer patients with some hemodialysis as well. And over time, because these are long-term catheters, these catheters get infected. And when they get infected, it's a nightmare for the patient, it's a nightmare for the treating physician, because there's a mortality risk there that can range all the way up to about 25%. In this case, a lot of these patients are severely immunocompromised, and as a result, something like this can be devastating to their continued treatment. So we think that And today the standard of care is that there really is nothing available. There's no drugs that are being used. The only standard of care is to remove the catheter and replace it with a new catheter. So now, how does a catheter-related bloodstream infection occur? One of three ways. One at the hub of the catheter, that's number one on the chart. And this is when either probably through improper hygiene, that hub is opened up, and from the skin of the caregiver, bacteria will get in, penetrate, and then the whole catheter is infected, or at the site of insertion, or hematologically from a distant infection, the patient comes down with a pneumonia or something like that, and that can backfill into the catheter as well and cause a catheter-related bloodstream infection. When that infection occurs, what happens is biofilm forms and forms very quickly. What biofilm is, it's bacterial growth, and ultimately it becomes bacterial slime inside the catheter and the lines. And there you see a micrograph of what it looks like. The bacteria have a real affinity for the plastic, and so they colonize rapidly in the catheter. So because these central venous catheters are a lifeline for cancer patients, they deliver their meds, they deliver their chemo, they deliver their nutrients. They're really critical to their continued existence. There are about 7 million catheters used annually in the United States. Close to 500,000 will be infected, leading to real life-threatening problems for these patients. As I highlighted before, the mortality and morbidity rates are significant here, and hospitals can actually be penalized for a hospital-acquired infection that becomes ultimately a catheter-related bloodstream infection. So, as I said earlier, the standard of care is to remove the catheter and replace it. So that is 2 separate surgical procedures, and those surgical procedures are very discomforting and very painful for the patient. There's a complication rate also associated with removing and replacing. Replacing with a new catheter, you have to surgically remove, usually the first time the catheter is placed subclavically by the collarbone. The second time when they have to remove that surgically, then the physicians will decide to put a new catheter in. Most of the time it's put in by the groin area and it gets threaded back up into the superior vena cava, to the heart area, which is where those catheters, the tip of the catheter goes. So the complications there they range from infectious, mechanical, and thrombotic, and there can be punctures that occur as they push that catheter up from the groin area into the heart area. As opposed to doing something with Mino-Lok when it becomes approved. There's nothing like this. This is unique. It will be unique in the marketplace. There's no other drug. There's nothing like this. What happens here is the nurse would come into the patient's room, unlock that hub, and inject our solution of Mino-Lok, which again is minocycline, sodium EDTA, alcohol, and that solution then goes into the catheter through the line and it gets locked. So it does not go into the human body. I want to emphasize this. It's very critical to know this, that this drug will stay and reside within the catheter and the catheter line. The drug will stay within that line for approximately 2 hours. At the end of 2 hours, the nurse comes back into the room, puts the needle in, and aspirates out the contents of the line and the catheter, and flushes the line, and the patient can then resume their IV treatment. So they still have 22 hours a day of uninterrupted IV flow. You do this procedure 5 to 7 days in a row, and that line will be completely 2 hours a day. The line will be, and the catheter will be completely sterilized at the end of that procedure. just to give you a contrast here, the cost of removing and replacing a catheter is about $10,000 in most hospitals. The cost of putting our Mino-Lok in is anticipated to be somewhere around probably $1,800 or a little bit above that. And this will be cost savings to the whole entire hospital system once this is approved. This, what you see here next is our phase 2B data, which was a trial that was conducted at MD Anderson with, as you can see here, the Mino-Lok arm was basically had no complications versus the control arm, where there was an overall complication rate of 18% in total. This is our phase 3 trial design. It's the protocol that's been approved by the FDA. The FDA really has encouraged us to get this drug approved because they recognize that it will be important for patients to have, and it will be life-saving in a lot of cases. So we are almost through this the clinical trial itself. The endpoint is time to catheter failure, and a difference between an antibiotic lock solution that the pharmacy will brew. I call it a home brew, and then as compared to Mino-Lok, and what the difference is in terms of catheter failure over time. So this is our development plan. As I highlighted earlier, we are in the late stages of phase 3. If we had not had the COVID-19 episode, the pandemic that's occurred here, we would have been done with this trial by now, and in all likelihood, we would have been under FDA review and well on our way to approval. But I'm sure I don't have to explain to everybody here, so this is a hospital-based trial, and that most of the hospitals did stop all their clinical trial programs when they were overwhelmed with patients coming in with COVID-19. What's remarkable about it is, though, we still have a small handful of hospitals that are recruiting patients. We get them monthly, a small number, but it's adding up. And our goal is that to get an NDA filed towards the back half of 2021, somewhere probably more likely the last quarter. But what we plan to do is, it's not our plan, it's basically what the Drug Monitoring Committee actually requested at the end of the last session that we had with them. They asked for a ad hoc meeting to be able to come in and look at the data again. If the statistics are powerful, there's a possibility, and I cannot predict this, and I don't want to predict this, but there is a possibility that the Drug Monitoring Committee could say to us, besides, they could say stop the trial because it would be unethical to continue because your data is that powerful. If that's the case, if that were to happen, then we'll get there a lot quicker, since we have a special status with the FDA called QIDP. This is just to give you an example. This is a worldwide opportunity. There's 4 million of these catheter-related bloodstream infections in the world on a worldwide basis. So we have intellectual property I described before, composition of matter patent that expires in 2024, but we have another patent that goes out into the mid-30s. But more importantly is what I addressed before. We have something called QIDP, and QIDP stands for Qualified Infectious Disease Product. Under this special status given by the FDA to antibiotic manufacturers and antibiotic developers to encourage antibiotic development, it's only awarded to breakthroughs that are considered to be breakthroughs. And under QIDP, our drug, once we file our new drug application, will be reviewed in 6 months instead of 12 months, as is normally done. And also, most importantly, we will get regulatory protection for additional time. So today, most drugs get, when they're approved under Hatch-Waxman, regardless of their patent status, they get about 3.5 to 5 years of approval. With QIDP, regulatory protection, meaning that no generic can come into the marketplace during that time frame. And again, if somebody has extra patent life, then as long as they don't get challenged, they'll have that time for protection also. But with QIDP, we get an additional 5 years, so we'll have close to 10 years of a protected runway, which nobody can come in, no generic can come into the market. And this is very critical to be able to have this type of protection. So we also have fast track, which is it expedites our reviews. So we have more frequent meetings with the FDA, as well as as we complete our sections of our filing, we can submit it to them for review. I want to highlight for you that at this time, in terms of the manufacturing side of our application and what we have everything just about completed on the manufacturing, or CMC as it's known. So we have lots manufactured. We've got stability data that's ongoing. All these we invested in a while back to make sure that when the drug approval comes, we're able to launch and provide drug to the marketplace. So as I highlighted before, there are no products being developed for the treatment of infected catheters. There is a company called CorMedix. CorMedix does have a product for for prevention of catheter-related bloodstream infections. These and they're being studied in hemodialysis patients. This is not in our something that we feel will be even remotely competitive with us. So just to highlight for you, Mino-Lok treats catheter-related bloodstream infections. It does penetrate that biofilm, eradicating the bacteria and saving that catheter. It's expected to be indicated as adjunctive treatment for the treatment of catheter-related bloodstream infections. And we have worldwide rights with the second patent that we have out there, the formulation patent, of almost 16 years of exclusivity. So this is a major step forward in addressing unmet medical needs. So let me move into novocyte mesenchymal stem cells, or IMSCs as we call them. Basically, we licensed this from a company called Novellus, which is a biotech company in Cambridge. They are unique in the sense that they have patented the only non-immunogenic messenger RNA product or therapeutic in the world. And this can be combined with gene editing and cell reprogramming. We have a worldwide license with them. The focus on it is acute respiratory distress syndrome. And this would represent the next generation of MSCs. The MSCs themselves, this is very technical at this point, but I'll just say it very quickly. They're derived from an iPSC, or induced pluripotent stem cell master cell bank. So this technology has significant advantages over others. One of them is that we don't have to go back to donors for the cells. So these are cells that are they're termed allogenic, meaning that they're off the shelf. So what we do is a single donor will donate a fibroblast. That fibroblast is then transfected 5 times using that special non-immunogenic messenger RNA cell reprogramming process, creating induced pluripotent stem cells. The pluripotent stem cells are then converted into a master cell bank, and that master cell bank, once it starts, will be able to produce cells on a perpetual basis. We never have to go back to generate any more cells. We can just keep using that same cell bank, and just keep growing cells all the time, over and over and over again. And this way, then you have consistency. It's predictable, and also what we have, we think, as an advantage over others, is that those cells have more potency than most of the cells that are being out there being researched. The cell reprogramming side, as you can see here, what happens is, what I'd like to highlight is that the IMSCs that we create are also known as being footprint-free, meaning there's no viruses, there's no viral vector in here. So we again have something very, very unique and very different. This describes what happens when you create the non-immunogenic messenger RNA. And this is the process how the iPSCs get created with the 6 days of or 5 times that the non-immunogenic mRNA is transfected or reprogrammed. So we have greater expansion potential than most of the other cells that are in the market are being researched, and we have greater expansion potential, as well as that we can restore telomeres, which is again something important in the cell therapy side. So this is something that is very unique. This is where medicine is heading. There's no doubt about it. This is changing the face of medicine as we speak. So this is an example of what that ARDS, what the COVID-19 virus looks like, and those spikes, if you can see them there, that are on the side. Those spikes have been described as being a killer with a crowbar breaking into human cells with impunity and injecting their RNA into the human cell, and that's why suddenly they divide and grow very rapidly in the human body. This shows you what the difference between a normal lung and an ARDS lung. I'm going to go through some of this quickly so as not to take up too much time. There is an immune response to the MSCs in these types of infections. This is the mechanisms of action for the MSCs in terms of what they do. And these are the levels of activity as far as the immune system is concerned. So what we're doing right now at this moment is we're underway. We'll be underway with a mouse study, and there's one mouse study that we did that showed the effect in mice with multiple sclerosis. We also have we reported on this a little while ago. We are conducting a sheep study or sheep model. Sheep, it turns out, have lungs that closely approximate the human lung, and what we're showing is that so far that our cells are producing therapeutic effects on those sheep lungs that have been infected. So our plan is we've submitted a pre-IND package to the FDA. Once we initiate and complete a toxicology trial, then we'll enter into man. Somewhere in the course of, we probably will be starting or getting ready for a human trial in the beginning of or end of 21 and beginning of 22. So this is still a little bit early. This could all change depending on what happens with further discussions and submission of data to the FDA. I just want to highlight this. It's more or less a very conservative point of view. So now let me take you into Mino-Wrap. This is a gelatin film, bioabsorbable, extended release antimicrobial wrap for the prevention of breast tissue expander infection. So what happens is following a breast mastectomy, if a woman desires to have an implant, what has to take place is her body has to be able to accommodate that implant, and something known as a tissue expander is implanted, and it gets built up over time to give that create that pocket. The difficulty sometimes is that in about 12 to 14% of the time, that tissue expander can get infected. When that tissue expander is infected, it can be deadly sometimes to the patient, but it's definitely devastating because you have to stop the chemo, you have to remove the tissue expander, and everything is put on hold, which is the last thing I think that a patient in that situation would want to have happen. This is what the wrap looks like. It's a wrap that has 2 antibiotics impregnated in there. That wrap gets wrapped around the tissue expander. It's put in at the time of surgery, and over a period of a couple weeks, it'll do what it's supposed to do, and then the tissue expander gets removed. So this is pre-IND at this point, and we basically are still a little ways off, but This product also will be unique in the sense that there is nothing out there like this. It's the only one of its kind, so it'll have the market to itself once we get an approval. So I'm going to switch gears from all the serious medications that I've just spoken about and talk a little bit about Citi-002, which is our hemorrhoid drug. Now, the reason this is here is because back several years ago when our company, which we had that was developing the Mino-Lok, we merged in with a public company called Citius, and Citius had a drug in its portfolio for hemorrhoids. We thought we weren't sure whether we wanted to keep it until we took a good look at it, and what we realized right away is that there was a significant opportunity here for us because there are no FDA-approved drugs for the treatment of hemorrhoids. So there are OTC preparations like Preparation H and others. Those are considered approved because they meet the over-the-counter guidelines for labeling and so forth. But on a prescription side, where there's a significant number of doctor visits and drugs being prescribed that are known as DESI drugs, meaning they were grandfathered in several years ago, or many years ago, I should say, when the FDA first got created. And they were allowed to stay on the market until companies came in, started doing clinical trials, and then these DESI categories disappeared one by one. That's what's going to happen here. There's still about $80 million worth of DESI drugs being prescribed in the U.S. market, and we think that once this is approved, this will be a nice market opportunity for others. This is not for us. We're going to take this through phase 2A, 2B, get some proof of concept in place, and then we're going to out-license this and monetize this asset for our shareholders. And we think, we know there's significant interest out there in mid-sized pharma and even big pharma for this type of drug. So basically, in terms of who we are and what we're all about, we have something that's really unique. We think that our company has great partnerships with both with MD Anderson as well as with the Novelis Biotech Company. Our portfolio does address unmet medical needs. We've got multiple near-term milestones. And again, this management team is a committed management team because of $26.5 million. So with that, I'm going to turn it over for questions from anybody.
Thanks for your presentation and participation, Leonard. I have a few questions. Some of them you received from the audience. So the first question, you disclose all the programs specifically for their developmental plans. I am curious if you could highlight the near-term catalysts, value-generating catalysts in the next 12 months for 2021.
Sure. The very near-term catalyst for us will be somewhere in the course of this year, hopefully within the first, towards the end, we believe towards the end of the first quarter, beginning of the second quarter, we're going to get a readout from our drug monitoring committee on a data readout on the Mino-Lok trial. That is a very critical readout for us, and that's the first series of events. After that will come, if they say continue with the trial, we continue with the trial. Somewhere in the course of the back half of the year, we'll be completing that trial, and then we'll be filing for an NDA. So we have many now near-term catalysts for the company, not counting what's going on with the cells and our dialogue and the work that we're doing there with the sheep study as well as other animal trials.
That sounds great. My second question, Leonard, is the therapeutic asset you acquired, you plan to start with ARDS. Do you have any strategies to tackle down other indications? If so, what are you going to focus on? Infectious disease? What would be your focus?
So our plan with the ARDS drug is to put it into a separate subsidiary, which we have done, called Novocyte. And that our plan is to fund that research without creating any dilution for the Citius shareholders. And that's about all I can say right now officially about that. I think there's different ways that you can create non-dilutive financing for something like this, especially if you have a separate subsidiary company, which is what we are doing, have done. So, and to focus on that company will be, we think there are other opportunities for us at Novelis for us to be able to in-license some other technologies as well. And we will take that one by one as we look at various opportunities that are there. But clearly, with that transaction that we have, that license agreement that we have something that moves the company from pharmaceutical land to biotechnology land. So this puts us in a different category altogether.
That's helpful, Leonard. My last question is your cash position and cash runway.
Say that again.
What is your current
burn rate? Okay, so right now at the end of our last published data came out of our 10K ending September 30th. We had approximately $14 million in cash on our balance sheet, and our burn rate is about, right now it's running about $1.4 million a month.
Okay. We actually received a question. So they're asking, you have one analyst covering you. Are you in conversations with other banks for additional analysts?
Yeah. So we do have two analysts actually covering us. One from Dawson James and the other one from Wainwright. Both analysts cover us. They do a really good job covering us. Yes, we are out there. We have Andrew Scott is in the marketplace, constantly making contact. And also we recognize that once we get to a data inflection point, that we are going to attract more institutions to the company. Because presently, right now, we don't have a significant number of shareholders or even number of shares that are represented by institutions. If you look at it, most of the shares are retail and ourselves. So myself and the management team, we probably have easily 35, 36% of the company, if not more. And so what happened was as we were funding the company, because we were such heavy participants, we really knocked out a number of the institutions that possibly wanted to come in.
Thank you, Leonard. Thanks for your participation and your presentation.
Hey, thank you very much. And once again, I appreciate everybody's participation, and I look forward, hopefully next year, at an in-person Noble company presentation.
Sounds great.
Okay.